Okay, now that you have your point selection tool, you're going to move down here and select the tip of each of the peaks. I'm going to move from left to right. You can see I get this little yellow point with the crosshatch on it. You can adjust its position using the arrow buttons on the keyboard until it reaches the very peak of your peak there. That corresponds to the center of the band on the gel. Next, we're going to go up here under the Analyze menu, press Measure, and you'll see this little window pops up. It's got a number of different parameters for that point. The one we're interested in is X. X refers to the position along this axis, left to right, on the gel. The units are pixels. So we're going to just go along here and select the top of each of our bands. And what I'm going to do is use the shortcut, which is Control M. Each time I press that, and I'm going to rearrange this window just a little bit so you can see both of them. You can see I've got an additional line of data over here. So I'm going to go along here and do this for each of the peaks and collect their X position along this plot. And it goes pretty quick. Just find them, and away you go. So now we've got a collection of our 10 standards moving from largest to smallest in this results window here. Now what we want to do is select all of these. Just take your pointer and drag down, copy, and we're going to manipulate these data in Excel. So fire up Excel, make that window nice and big, and we're going to open the Excel worksheet that I've generated for you guys. You can download it from Angel. You'll notice it's an XLSM. The M refers to the fact that there are macros embedded in this worksheet. The first time you open it, it will ask you, do you want to enable macros? You select yes, or enable macros, otherwise the sheet will not work. These cells where you see data has already been entered are locked, and so that will prevent you from disturbing the function of the sheet. If for some reason you need to unprotect it, the password is down here. It's just fergplex, but I see no reason why you would need to do that. So we've copied the data from this results window in ImageJ, and now we need to paste it. This is just going to be our short-term pasting area, so you press paste. The only information that we're interested in is this row, or excuse me, this column. This column is the X position on that plot. So you're going to copy that, and we're going to put it up here into this region. I'm going to zoom this up just a little bit so we can see all the parts that we need to see. This molecular weight standard region of the sheet here, what we're looking at is the observed pixel position. These are the data that you just recorded. This column is the known base pairs based on the way the ladder was produced. So this is the exact base pair number. And what we'll do is just paste those data right into this column right here. And so these are the positions, and what it's doing is it's matching up the known base pair size with the observed pixel position. So if that makes sense, what it's doing is correlating these values, and if we scroll down a little bit here, it's going to plot these on this plot where on the y-axis we have size in base pairs, and on the x-axis we have the position along the plot, or the position in the gel. And what we're going to do over here is for each lane we're going to enter in data for the peaks that we see for each of the loci. You see here there's the name of the locus. In the red channel we have 5, FGA, TPOX, this STR, BW8, and the gender determination amelogenin. And then in the green channel we have 6 additional bands. What we're going to do is go back to ImageJ and collect first the red samples and then the green samples. So we're going to go back to our plot. We're also going to bring up the results window. so that there's not too much data in this results window. I usually like to clear all these data so that we don't have to wonder, oh, is that the latter or is this the unknown? So let's scroll down here. And first we're going to collect the red data. So go along here and with your little point selector, Control M, Control M, Control M. And you can see it's populating the results window over here with that data. There we go. So we've got 1, 2, 3, 4, 5, 6, 7, 8 peaks. If we look over here, we can see we have 1, 2, 3, 4, 5 different loci. Because we have 5 loci, we would expect 10 peaks. That indicates that at least some of these loci are homozygous. If we go to our gel, we'll see if that makes sense. So if we look in this red channel, bring up my ImageJ window here, get the hand. Moving from the top, you can see this locus up here, which is FGA, the largest one. We have 2 red bands, so we have heterozygosity there. 2 bands at the TPOX locus. But when we get here, you can see this yellow band. There's only a single red band underneath here. If we go to color and composite, and we actually turn off the green channel so it's a little less confusing, you can see that there's only a single band here. So this individual is homozygous at this locus. They're also homozygous at this locus. And I'll show you how to deal with that whenever we get over to the Excel spreadsheet. Let's select our data again. It'd be nice if you could just select the X column, but it doesn't appear that you can. Copy, go back to Excel, and once again, this is just our pasting dumping ground here, so we'll paste that data here. Copy, and paste. If you want to maintain the formatting here, just click this little guy here and just select paste values, and that will prevent you from losing any of the formatting in here. Now, it's nice to look at the gel right alongside whenever we're doing these analyses, so that we can determine, well, which loci are homozygous and which are heterozygous. As we said, we've got 2 bands up here for FGA. Okay, that's these pixels and these pixels. The formula in the Excel sheet is interpolating a pixel position of 313 as base pair 357.7. Now, there's a formula over here that says, okay, let's look at this predicted base pair position and go over here to this table of alleles and their corresponding base pairs and find the closest base pair number and return the allele that corresponds to that. So you can see 357.7, it's predicting 25, and 358 is the appropriate base number. So we're about 0.3 base pairs away from what it's returning here. Now, as we said, these 2 values correspond to these 2 peaks, so that's FGA. These 2 peaks correspond to TPOX, but then we're homozygous for D8S1179. So that means that this is the only allele present, and that these numbers here correspond to the next set down. So what you can do is just Control X, and then we're going to paste down here. I screwed up the formatting, but for the purposes here, it shouldn't matter. You can see that this is automatically defaulting the blank cell to the highest allele. You can just copy this and paste it here because these 2 bands are running right on top of each other. What that means is that there's only a single band present at this locus. It's homozygous, and so the genotype is 1515. The maternal allele is 15 and the paternal allele is 15. If we go down to the next locus, right here, which is VWA. You can see that we also have a single peak. So that means that this is the appropriate peak for that. And we should have two of those, so we're going to shift these down again. And then copy this into here. So VWA is going to be homozygous for 16. And then we have the amelogenin locus. And this one would be easy enough to interpret by eye. See, it has two bands, the larger band corresponding to the Y, the smaller band corresponding to the X. Excel has predicted that appropriately to indicate that this is a male. So what we see is that at the FGA locus, we're heterozygous for 25 and 20 repeats, TPOX heterozygous for 11 and 8 repeats, D8S 1179 homozygous for 15, VWA homozygous for 16, and we have a male. So now we're going to have to do the same thing for the green bands. If we go back to our plot here, bring it up, bring up our results window. This is going to be treated exactly the same, and this will be a nice review so that you can feel confident in your analysis. So go into here, clear our results window, and we need to go back to the ImageJ window, select the point selector, and then move right along and Control M. Okay, we've selected all of these green peaks. Let's go over here, copy, and back to Excel. Our pasting dumping ground. Pick up our X values, copy, pop them right in here. For some reason we have too many. Huh. Okay, I see what's happened here. We have an extra cell here. If we go back to our gel and our plot, you can see we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 peaks, plus this 13th peak here. Because we have 6 loci, that should correspond to 12 alleles. The 13th band, if we look on the gel, is this band down here. This band is either non-specific amplification product or a large primer. I believe it's a non-specific amplification product, but it should not be counted. And in fact, you see it in every lane here, including this lane here, which is a negative control. So it is clearly not an actual amplification product. So what we can safely do is go back here to our Excel sheet, and we're going to delete the spurious data point down here. And these are observed pixels. Once again, using our known molecular weight standards and a cubic spline interpolation formula, which interpolates point to point between the molecular weight standards, we're predicting a molecular weight for those bands, and at each of these loci we're returning a value that is the closest repeat number to this base pair. This is based on data present over here in these tables. These tables have been retrieved from STR base. So this is how we retrieve a specific genotype for a given individual. Each of you will be determining the genotypes for several individuals. And if you look at the workbook here, we have several worksheets at the bottom. And so as we go to the bottom of the Excel worksheet, Here's sample 1, sample 2, sample 3, sample 4, and so on. What you can do is rename this sample to the specific identity of the sample that you're analyzing. So you can change this to whatever it is that your identity is that you're testing. So this is how we generate a specific profile for an individual, and we'll get into the statistical analysis of allele frequencies later in the lab.